Okay. Hello once again, everyone, and welcome back to Bioinformatics for Infectious Diseases. This session is session number 6, and we'll talk about disease progression and outcomes, continuing from where we left off last time. Last time we talked about the differences between genomic sequences of Ebola virus and focused on the glycoprotein. And then we also started talking about the glycoprotein in the SARS-CoV-2 and the coronavirus. So we'll continue to talk about the different symptoms caused by viral proteins and in the context of viral replication and immune evasion. So we continue to talk about those examples. Before we start going through the material, let's review some of the things that I've been kind of taking a look at today to catch up and see where everybody is at. So as you know, we have the main page. This is the link. I'm actually going to just paste this link in chat for all of you to see. Okay. Okay, one second. I'll paste this to everyone. Okay, so on this page, we track progress, and that's why I was asking, how everyone is doing, because I think it's important not to kind of lose everyone with things that we start doing, and they might be something that you haven't really had a chance to try yourself. So I wanted to take the opportunity to review where everyone is at. We have a couple of people that have been making more progress than the rest. So congratulations to Roland and Renine. I want to encourage everybody else who joined the program to start looking at some of the material that we have and do some of the pipelines so that you get a sense of how this data is analyzed. And if you have any questions, we post the recording sessions from previous meetings that we had. So all of the meetings here should be up to date. We have session 1, 2, 3, 4, and 5. So those 5 recordings are there. You can also look on the forum in some other areas where we have questions. If you think that something's not working or have a specific question about something, you can ask here. For example, I saw there was a question. Let's see where it was. Okay, right here, there was a discussion. So this is under assignments and resources. I pasted here some of the links, including some of the coursework, some of the resources about virology and genomics, if you're interested, a video on the Ebola virus virology. And then also someone was asking about download FASTA files and opening them. So I wasn't sure what exactly the question was, but I did paste a video on how to go to NCBI virus, find sequences, and download them to your computer. Then in one of the sessions, we discussed a number of different software tools that you can use at home on your computer to get a better sense of the outputs, including the alignment of different genomes to see how they are different, and visualization of phylogenetic trees. And then I posted here some of the publications that we discussed, and there was a question about creating a pipeline to start multiple sequence alignments. So Paris was talking about phylogenetic trees on NCBI virus and how to use it on your profile. So the main point of the pipelines is to understand how the analysis works. And to do that, I put here a number of steps, and we'll review those today on how exactly you would go about adding those FASTA files into the pipeline, running the pipeline, and then looking at the output, for example, using some of these tools that are described up here. So if Paris is here, hopefully that makes sense. If not, maybe in the video, you'll be able to review that. Again, I also added the different kind of topics we discussed, right? So we asked that in the first session, you complete your profile, make sure that you have access to everything. Then in session 2, we started talking about NCBI virus, and we produced a multiple sequence alignment of the different Ebola viruses. We talked about mutations, and so I posed some of these questions. These questions are to help you think about the topics that we cover in the session, hopefully to make this session, more relevant, because if this is completely new and you don't have the time in between the sessions to review some of this material, it really, I think, requires a lot more explanation than we're able to fit into this 1-hour session. So here's the last session's discussion that we had, which was to read this publication on the structure, function, and antigenicity of the SARS-CoV-2 spike glycoprotein, and we'll discuss that today. So hopefully you had a chance to see that. And then I did just review some of the top performers, like I said. So Roland is way ahead of everybody else. Congratulations on completing all of this coursework. And then Winnie was also completing some coursework and starting to run some pipelines. So that was great to see as well. And if you have any questions, I highly recommend you post those questions here. That way we have enough time to review and prepare the answer for the session itself. Okay, so I do want to mention that we will have a free webinar for those that are new to all of this and would like to understand better how to deal with 3D structures of proteins, how to load them, visualize them, understand the relationship between genetic or genomic sequence and the function of the protein. So there will be a full program. The program is going to be led by Dr. Sona Vasudeva, who is a faculty at Georgetown University Medical Center. So that program will start on July 7th or 8th, I believe. If you're interested, it's going to be an online program. It's not specific to Louisiana Biomedical Research Network, but it is designed for beginners. Those are just getting into all of this. And we have another program also for beginners, even though I think the topics there are quite advanced. They're going to be focused on data science. And those are also will start on July 8th, a full program with iResearch Institute and Science Coach, 2 organizations that have a lot of impact on research projects developed by students. And now in the time of COVID, no one has access to any labs, so this is kind of like a dry lab experience. So if you want to hear about these programs and get a little bit of a better sense about protein and genomic sequence relationship, kind of genotype-phenotype relationship at the protein level, and are interested in data science and coding and data visualization, you're welcome to join our session on July 1st. Let me share that link. If all you need to do to receive the invitation is to submit your information on this link. And we'll get back to you with a Zoom link for July 1st at 10 a.m. CST. So today we'll talk about the glycoprotein in SARS-2. But before we jump into that, let's review what we covered in our last session. We talked about the evolution of Ebola virus. There was a publication that I asked you to take a look at, which was the evolution of Ebola virus insights from the 2013-2016 epidemic. And I don't know if any of you had a chance to read that publication or look at it. maybe we can spend just a brief couple of moments talking about that publication. Did anyone read that publication or take a look at it? Okay, Maya, do you remember what were some of the main topics that they focused on when they were analyzing this data?
So what I remember from reading it was that they were basically talking about how um, there was like a glycoprotein like mutant that contributed to like more deaths, and they were trying to figure out like, I don't know, like how to analyze that and when it came in.
Yeah. Okay, great. Yeah, so that's exactly what they were trying to do. They were looking at that glycoprotein for multiple reasons, right? So one of the reasons was because they were concerned about the source, the origin of the epidemic. And for the origin to be more dangerous, there must be a presence of the exact same sequence, right? So this glycoprotein is responsible for the entry into the human cell. It has to have a certain affinity to the human receptor. And for it to enter into the human cell from an animal reservoir, from what's called the zoonotic spillover, that event can either happen once, and that means that some virus in some specialized location in one rare instance jumped over between an animal and a human. For example, a human was, out there in the middle of the jungle, cutting down trees, got cut, and at the same time, there was virus present from some non-human source. So that interaction is a spillover event. If that same virus with high affinity to human receptors in the glycoprotein is circulating in the wild, and that is happening in close proximity to the human population, there's more concern about continuous spillover, and that makes it more dangerous. So that was one focus. The other focus was on specific mutations in the glycoprotein that have contributed to the virus spreading more rapidly or being more deadly, so causing more deaths. And that concern was the focus of a study where they looked at the number of infections, so infection rate, and the number of deaths, mortality rate. And we're trying to associate those two factors with the genomic sequences of Ebola viruses identified in those patients, specifically looking at this glycoprotein. And two conclusions from that study. The first one was that this was a one-time spillover event because you could trace the genomic sequences back to a single origin. However, when they looked at the prevalence of Ebola viruses in bats, they've identified a specific species of bats that lives right around the area where these continuous spillover events happen. And we see that there's another outbreak of Ebola virus that was just recently contained. So this might be a bigger concern going forward. On the other hand, about that individual spike glycoprotein and mutations associated with more deadly outcomes for patients, they looked at the glycoprotein. By the way, these kinds of studies really helped advance our understanding of Ebola virus biology and brought the capability to design vaccines and antiviral drugs. But in the glycoprotein, that mutation that was associated with higher death rate really, it seemed like, had more impact on how many people were getting infected. And so maybe it's more of a transmission rate rather than fatality rate. By the way, does anyone know what is the fatality rate in Ebola virus? you can even guess. What do you think it would be? 90%, 50%, or 20%?
I'd say about 90.
Okay.
I say 50, maybe.
Okay.
75%.
So I think the latest is that it's about 50%, which is very high. Who knows what's the fatality rate or mortality rate from coronavirus, this novel SARS-2 coronavirus?
Is it like less than 10? I don't think it was, well, now it's like mutated, but before the mortality rate wasn't that high.
Okay, so yeah, that's true, but let's say how many? So was it 15, 10, or 5?
I'd say 10.
Okay,
I'd say 15.
okay. Anyone else? Right, well, I think it's closer to 5% now. You know, that makes it about 10 times less fatal than Ebola virus. And just as a comparison to, let's say, influenza, about 2 to 3 times higher than influenza or flu. So those are some of the numbers just to give us kind of a perspective. Ebola virus, one of the biggest concerns throughout the Ebola virus outbreak of 2013-2016 was that potentially that transmission rate could be so rapid that many, many more people are going to get infected, therefore many more deaths, and easier to spread in new locations where populations had zero interaction with those types of viruses. So their immunity was not ready for that. And so that's maybe another aspect of why the mortality rate might go up. So those are some of the concerns with the Ebola virus. What's interesting is that the same glycoprotein, it's a different protein, but it's the same function, so to say. You have here, yeah, Deidre, did you want to say something?
Yes, I'm sorry. Yes. Well, let's say when there's an initial infection with the virus, that rate can change, right, over time. So let's say initially when, let's say COVID came out, was it that low? Was it just 5%?
Right, so that's a great point. We talked about how the initial spillover event, because it changes between different environments completely and adapts to a new type of a host, that mutation rate can be much faster, right? The mutation rate. The mutation rate means that the virus is getting adapted. And the rate of mortality, right? How many people die out of those that are infected, it's a little bit of a different story. And I think that one of the challenges when an early outbreak happens is that there are so many cases that are not tested. So truthfully speaking, we probably don't know. And we will know a lot more after the outbreak is over and people have the time to process all the data in a very rigorous way and really account for all of the factors. The fact that it is spread in so many countries and so many different groups are doing the testing in so many different ways makes it very difficult to universally say what that rate is globally, right? So I think once everything kind of calms down and there's a systematic review of patients that are not, biased by any other tactics like government suppression of numbers or on the other hand, encouragement to, get to the bottom of what exactly did every person have, right? Those factors play a huge role. So I'm not going to speculate. I would say that, I think that rate around 5%, that number is likely going to change. How long and how much data do we need to really establish that rate? I think we'll have to see. So the point though is that, they use data analysis, right? And that's what the program is about. bioinformatics. And I think that it's very important that we not think about these subjects just as a matter of experts having an opinion, or government agencies putting out some numbers. I think that the most reliable way for us to look at it is to really learn from the different analyses that experts have done and try to replicate the same analysis to see whether we can identify similar trends that they have seen in data that has been published. So we continue to talk about the origins of SARS-CoV-2 and this novel coronavirus. What was, one of the biggest topics in the early discussions was, where did it come from? Because as opposed to SARS-1 or MERS or even Ebola virus, that same exact sequence, the genomic sequence of that virus, has not been found in any other animal. And that's why a lot of people had these questions. Well, where did it come from, and what is the evidence that it was that it occurred in the market, wherever they said, or any other place, right? So then a number of publications came out, and this was one of the publications that we started looking at that talked about some of the evidence that they found in specific genomic sequences. You can see those genomic sequences right here: the novel coronavirus in human, the closest coronavirus in bats, a pangolin coronavirus, and then some of the older coronaviruses in humans and in bats. Because if you have a coronavirus that is diverse and present in bat species and has caused a pandemic in the past, like SARS-1, you would assume that something very similar would be happening again. And when they analyzed those sequences, one of the things that we started talking about was a specific insertion in one of the regions of the glycoprotein of the novel coronavirus. And it's interesting because, again, it's the glycoprotein. So we already learned a little bit about the structure and function of the glycoprotein in Ebola. Here we have a different protein and a different virus, but also the difference between a previous outbreak and this outbreak specifically is in this spike glycoprotein. So I wanted to repeat some of the steps that we went over up until now for multiple sequence alignment, right? So what are the steps that actually allow us to align genomic sequences, and where does the data come from, and how do we actually get it to this level? If you can see, I don't know how big the screen is for what you're looking at, but here we have the genomic sequence of multiple viruses. I don't know, does anyone see what the letters here are? They're G, A, G, I, C, A, S, Y, G, or Q, T. Okay, so what are those letters? Are those nucleotides or amino acids?
They have to be amino acids because nucleotides are only like I forget what it is, but it's not Q. That's all I know.
Exactly, yeah. So nucleotides are T, C, G, and A. And here we have all of these letters Q and Y and so forth. So these are nucleotide sequences. And so I wanted to go over again the steps you would need to do to get to the same result. Alignment. So first of all, finding the data, right? Then alignment, then conversion to amino acids to get this kind of a comparison. So let's review, and then I want to show you just an example because that was one of the questions in the forum. So number one, you have to go to NCBI virus. And NCBI virus allows you to download FASTA files. Two, once you've downloaded the FASTA files for the sequences you want to study, you have to have a reference genome. The reference genome is not only to align to compare with the reference genome, it's also to have the codon sequences, right? Because each trinucleotide sequences form a codon, and the codon is actually what codes for amino acids. So to know that conversion, we have to have a reference genome, and that will be on NCBI. Then number 3, we actually have to go to the platform and perform phylogenetic analysis. So phylogenetic analysis could be done in these 2 sections: virus quasi-species. Yeah. Did you want to say something? Okay. So, you know, going on the platform, you're able to do this kind of analysis in this phylogenetic tree or in the virus quasi-species. We kind of talked about that a little bit last time, a section dedicated to multiple types of analyses that are useful for viral and next generation sequencing data. After that, you have to upload the files and sometimes organize them into groups. So it will be, you know, when you have a lot of files, a lot of FASTA files, it becomes difficult to understand which one is which. So organizing them into groups might be helpful. So here I have an example, and some of those files were shared with all of you: beta coronaviruses, COVID-19, MERS, and SARS, and we can look at comparisons between them. Now, what would the pipeline look like? The pipeline would have loaded those files, then perform multiple sequence alignment at the nucleotide level, converts them to amino acid level, and then uses only specific portions of the codon to build a phylogenetic tree. In fact, this is not a necessary step, but if you do it, you get the full table. Okay, so let's take a look at, and then build phylogenetic tree. You can either use one of these or do a comparison between multiple types of phylogenetic methods. And then you can analyze those different methods, comparing them to each other and finding the kind of average tree between multiple methods. Okay, so let me ask you this. How many of you were able to build this kind of a pipeline and run it?
I was able to do my pipeline with Ebola and with Corona.
Okay, great. Did anyone else?
I did one with all of the beta coronavirus, SARS, MERS, all of those.
Okay, and
I did
yeah.
as well.
Okay, and did you happen to look at the results?
No, I did not. I don't think I did my phylogenetic tree. I think I missed that step. I'm not sure.
Okay. Did anyone else look at their results?
I haven't yet, but I can look right now.
Okay, so please do. As you're looking, let's all take a look at one of these pipelines. So again, the pipeline that we're going to look at the outputs from is a comparison between multiple types of coronaviruses. And here I have actually a second one. So the first one that we discussed already is a comparison of human coronaviruses. But then we started another one that was a comparison between the same types of sequences: bat coronaviruses, pangolin coronaviruses, and human coronaviruses. So let's take a look at what some of these outputs are. So I'll go to, you know, this is a pipeline that I did, but you're welcome to look at the outputs as well. So you can see, if you haven't run a pipeline, you should be able to see the same results. So here you can see a bunch of different outputs. Some of them are simple, like you can just look at this PDF file. Let's take a look at what this PDF file will show us. Okay, so here's that PDF file. And what is good about this PDF file is that you can see that everything was organized by group. And so it's easy to see that, here's all of group 4, here's all of group 1, here's all of group 2, etc. So you can start looking at how the groups are related to each other, right? And this phylogenetic tree actually shows us their evolutionary relationship between them. What other information do we have here? We have FASTA files. So the FASTA files are the alignment between those different FASTA sequences that we uploaded. You can actually look at nucleotide per nucleotide alignment. So if you're interested to find differences, you'll be able to spot them right away. And there are some other ones, including this clustal. Clustal is also an alignment file that you can visualize, so you can see the differences between those sequences. Now, why are there so many different outputs? Because it does one time for all of them together, and then another time it does it individually within each group. So if there are a lot of differences between different groups, within a group, the alignment might be better, and you might be interested to know what are the differences inside each group. So that is another benefit. But there's a central table here that is called MSA, bat, pangolin, human, SARS-like full table. So this full table, I'm just going to open this table together with you. And let's take a look at what is inside. So these are the different groups that we have. You can see that they are annotated. SARS-1, right? These are the actual accession numbers. This is our reference. So this reference is the reference genome. What is this genome? We can take a look. Okay, so you can see that this is a reference genome for the original published Wuhan HU1 complete genome that was published by the Wuhan Institute of Virology. And that means that we are aligning everything in reference to that SARS-2 genome. Now, also you have SARS-1, bat SARS-2, bat SARS-1, SARS-1 in humans, bat SARS-1 in humans, COVID-19, pangolin. Okay, so this shows us how we have a number of different groups, and within each group we have a number of different genomic sequences. And this is just nucleotide sequences. When you originally download that file, it actually has a lot more. So there's some nucleotide sequences. If you scroll down, you can see how each nucleotide sequence is converted into an amino acid, right? So there's all of those letters, D, S, V, etc. So for each sequence, you have a nucleotide sequence, an amino acid sequence, and since the same approaches could be used for DNA, where you have multiple two strands, negative and positive strands, you also have additional, so we don't actually need these three. But here you have the nucleotide sequence and the amino acid sequence. And what we can do by looking at this data, we can actually extract just, for example, here we have extracted just the relevant ones, right? And then one of the first things that I can do is organize them by group and look at similarity. So how do we look by similarity? This is a manual exercise for you to understand kind of how people produce these kinds of charts. You can see that in total, we can calculate how many we have, right? So in each position, we calculate how many we have, and then we can look at whether a specific sequence, for example, if we have this symbol, it means that there's a nucleotide missing there, right? If there is a nucleotide, then it's present. So we can start calculating similarity out of total that we have, how many do we have in each one and how many are exactly the same, right? So here we can produce, as a result, a plot of similarity of these sequences. And here each line represents one of those groups. So you can see that this is bat SARS-1. This is the blue one is pangolin. And the red one is average. And the yellow one is COVID-19, right? So we added those right here. Overall similarity, pangolin, COVID-19, and bat SARS-1. So just an example of from 40% to 100%. Yes, Deidre, did you want to ask a question? Did you want to ask a question? You can go ahead.
Oh, I'm sorry, I didn't unmute. Okay, were those the figures on the graph automatically generated, or is that something that do we have to actually do something in order to generate this particular graph?
Great question. So this graph here is generated in Excel, but there are a number of tools that you can use to generate these kinds of visuals. Here are some of those tools. I've added them, and I will add them to the forum if you would like to use them. There's a tool, one of these tools is in R. You can, there's a package in R that allows you to generate all kinds of plots like this. There's also a tool in Windows that you can use to generate these plots. I just, I think it's a fairly simple plot to generate, and so you can do it in Excel. But if you wish to use one of those tools, I'll post them on the forum.
Okay, thank you.
Okay, so let's take a look. We have from 40% to 100% similarity scores for each one of the groups. Where do you think, right? So overall, I think it's fairly obvious that the pangolin, the blue line, and the pink line are comparable, right? So they go through, and the majority of the time, right, both of them are right around this red line. In some cases, some of these lines actually go down, right? So here you can see the pangolin similarity goes down and the bat similarity goes up. So that is an indication that in this specific region, right, and we can see down here what region that is, pangolin is less similar to the human COVID-19 than the bat sequence, and so forth, right? So we can go through, and you can see that there are specific dips in only some regions, right? So those are the dips that we are interested in. Since this is a nucleotide sequence, this just gives us a sense of nucleotide variability, but we can build the exact same plot using the amino acid sequence just based on this one output, which is the table, right? So again, where is this table found? This table is found right here. Deidre, did you have another question or?
No, not at this time.
Okay, just see that your hand is raised. Okay, so that is where you can get a plot, a similarity plot, for example. We talked already about some of these other tools that might be useful for visualization of the FASTA files. We saw the PDF file, and then an additional one is this birth death process.trees. So what is this format.trees? I showed you how previously you can use a something called Figtree, a software called Figtree. So this is what Figtree looks like. And we can open this .tree format in Figtree and actually add a lot more information to the actual phylogenetic tree. What kind of information can we add? Well, obviously we can annotate the clades. And if we start annotating those clades, you will see here how we see MERS group standing here apart from everything else, which potentially means that it's very dissimilar from the rest of them, right? And you can see that if we think of this as our time scale, it is the farthest away, connected to everybody else at the source. So potentially they had some common origin because they're all coronaviruses, but they're not really that closely related. The next group that we have here is bat SARS-1, but inside we also have SARS-1 the human, right? So the bat SARS-1 human and the SARS-1 human are closely related, and you can see them together in this group. Here we have the pangolin, and here we have the pangolin. But in between this pangolin group and these two, we have the bat SARS-CoV-2 C45, one of the sequences from the bat SARS-CoV-2. So this is a sequence that is more closely related to the novel coronavirus in bats than the other SARS-1 bat coronaviruses. And here you have another bat sequence called bat rat G13. Remember, we looked at that file already. So this sequence and these two pangolin sequences in general over the whole genome are closely related to the novel coronaviruses in humans. So the phylogenetic tree here is a format in .tree format that allows us to modify how the tree looks and add all kinds of annotations. If we look at this graph, where do you think is the largest, the longest region of dissimilarity? How would we know? How would we know which region in the genome has the most dissimilarity? Does anyone want to try and answer that?
Do you look at the peaks? Do you look at the peaks?
Okay, so the peaks, you mean the ones going up?
Yes, sir.
Well, this is the percentage of similarity, right? So 100% means it's identical. This is COVID-19 sequences. And in between that whole group of COVID-19 and humans, there's very little variation. You can see it's all around 100%. So the lower you go, right, the more are they dissimilar. So if they are more dissimilar, which region on the genome would you say has the most dissimilarity? What do you think, Roland?
Which region?
Yeah. How would we go about finding that out?
Like the most similarity would mean like that their peaks are touching each other. So I would say region 3729 is the one that's closely related, because like they're stacked on top of each other.
Right here is what you mean, right?
Yes, sir.
Right. So I was asking, right? So how are they dissimilar? Meaning, how are these two, pangolin and bat, most different from the COVID? So this is correct. This is one region where there's a lot of differences between both bat and pangolin and the human, but between themselves, they're very close. So you are correct there. Deidre, did you want to add something?
Well, I was about to say that would be the peak I would choose.
Okay, great. But let's look, right? What does dissimilarity mean? Dissimilarity, in fact, does mean how many are different. And this height represents, in this particular region, how many are dissimilar. But what about longer regions? You can see right here, a fairly long region, right? It might not be as low, but it's long from this reference genome. So the question that I have is what other information do you need to tell me which region of the genome, what kinds of regions does the bat coronavirus or the human coronavirus have? What are those regions called?
Hypervariable.
Okay, hypervariable regions are hypervariable regions in bacterial 16S ribosomal RNA. That's true. There is a thing like that. But in this specific situation, we need to know the genes, right? So every genome is organized into elements, and those elements, you know, the very basic ones are genes. So here are the genes for this coronavirus. You can see that there are ORF1A and ORF1B, and then S stands for the spike glycoprotein, and then you have these other proteins right here, right? So this region right here is actually the longest region of dissimilarity between the pangolin and the bat. Between pangolin, bat, and human. So looking at this, can you tell me if the bat or the pangolin is more closely related to the human sequence, specifically in the spike glycoprotein? Which one is it, the bat or the pangolin? The bat is pink, the pangolin is blue.
The pangolin.
Yes, the penguin.
Pink one or the pangolin one? Because the pink one is bat and the blue one is pangolin.
So you're saying which one is most similar?
Yeah, more similar.
More similar to the initial one. Yeah, the yellow. Yeah, and I would still say out of that group, I guess that would be the blue, the pangolin.
Okay,
Yeah,
excellent. Right, Roland, would you agree with that?
Yeah, I would agree with it because it's close. It's got a longer peak.
Okay, so this is interesting, right? Because overall, we think of the main origin being bats, but specifically pangolin was proposed as one of the intermediate hosts, right? And that's why we're doing this analysis to kind of see which one is more similar. And we know that the spike glycoprotein is the entry point into the host cell. The hosts are different, right? So the human, pangolin, and bat coronaviruses have different receptors. And now we are trying to understand, okay, well, if for it to cross over, it needs to be fairly similar, right? The more dissimilar it is, the more difficult it is for it to get in. So our analysis shows some interesting pattern within the spike glycoprotein in reference to where these coronaviruses might have originated from. And here's indeed a publication where the exact same comparison is being made. You can see now different types of genomes here from bats, SARS-CoV, and others. And this is that same region, right? So that region that we saw, which is right here, and you can see this dip down here. Now, you can use, do some of these tools, but today we wanted to start looking at why that is important. So what is going on inside this region, and how do we interpret this dissimilarity or similarity further? So I asked you last time to take a look at this publication. This publication talked about this glycoprotein. Did anyone have a chance to read that publication?
No.
Okay, so if none of you have had a chance to look at it, please try to go back and read through. I think there's a lot of good information in there that will help us continue explore the role of this glycoprotein. But let's take a look. So here you can see this is that spike glycoprotein. And we want to understand, how does this similarity or dissimilarity between these different sequences actually could be interpreted in terms of how a pangolin might be the intermediate host, and what does it mean for the virus to go between two different hosts. So let's look at where we got this data. So last time we talked about going to NCBI and downloading these data sets, right? And you can basically do that selection yourself. So just to remind everybody how to do that, I created a video that I also posted on the forum. If you would like to, and I would recommend for you to do this, to go back and redo this analysis for yourself, that would be very helpful. Once you do that, remember that you have the genomic sequence aligned to other genomic sequences. And the genomic sequence codes for different genes that eventually become proteins, right? And so when we think of how to understand the meaning of some dissimilarity or similarity between these different genomes, we need to understand what impact do they have on proteins. Now, what does the virus actually do? The virus, the coronavirus, when it enters into the cell through a process called endocytosis, what it does is there's a spike glycoprotein here covered in green that connects to an ACE2 receptor. And that receptor actually takes off a portion of this glycoprotein, which then transforms and fuses between the viral membrane and the cell, the host cell membrane. And that is that critical entry point where the viral RNA is then inserted into the human cell, because the virus on its own cannot replicate. So when we talk about disease, when we talk about infection, that infection is the result of the virus being able to enter into your cells. Not all cells have the same receptors. Not all cells have as many of these ACE2 receptors. So how do we know protein function? For example, how did people come up with this diagram? There are several ways that you can think of protein function. Protein function could be studied by similarity. When we do alignment, let's say we got some region on the genome and we want to understand what does this region of the genome stands for. And we can analyze it by comparing to other known sequences that are similar to this genome, to this portion of the genome. And we can look at how they might do similar functions because of their similarity. Another way that we can look at them is by looking at their 3D function structure, because the genome is on an RNA. That RNA, once it is translated into amino acids, it folds. And this three-dimensional structure, the folded structure, could be studied. So those could be studied again by microscopy. So cryo-EM microscopy can reveal the full structure of that protein, or it can also reveal the protein bound to another protein. And so we will look today at this aspect of this protein function. So we can find this spike glycoprotein on a database called Protein Data Bank. And specifically, you can see here structure of the SARS-CoV-2 spike glycoprotein closed state. There are two stages to this protein. It can be either closed or open. And you can see here a representation of the virus and the spike glycoprotein. And this is the view from the top. So you can see this enlarged area here on top. This is the view from the top. So let's go and take a look at this resource. And I'll paste this in chat as well for you to take a look. Again, this site is called Protein Data Bank, or PDB. So you can see here, you can also look at 3D view. Okay, they have some kind of an unavailable service. Okay, there we go. So here you can see the sequence, right? This is the sequence in, what do you think this is? Is this nucleotides or amino acids?
Still gonna go with amino acid. Wait. Nucleotides.
Okay, and why would you say that?
Because of I say nucleotides because I say nucleotides because if you look at it, the SARS corona, like it's kind of complementary to one another, and the numbers 212, 222, 232, 242, it's like in a pattern in a way, like up by 10. Like it's going up by 10, and like wherever the patterns are, that just shows like where the nucleotides are present.
Okay, so anyone else want to comment on the sequence? What is this, a nucleotide or an amino acid sequence?
I say amino acid.
Amino acid.
Amino acids, I think.
All right, I agree. This is amino acid. So Roland, how do we know this is amino acid? We know because there's only 4 nucleotides, T, C, G, and A. And once you start seeing these, WTF, that tells you that this is amino acid and not nucleotide. So when we look at a protein sequence, and this is a protein, we would typically think of it in amino acids. When we look at the genome, we would think of nucleotides. Okay, so here's the structure. You can play around with it and just kind of spin around, but if you point it upwards, you can see how it forms this kind of a structure like a spike, right? It's kind of wider here on top. So this sequence is actually encoded by the gene S, and this gene, or this protein, once it's produced, has a few different functional domains. And these functional domains are organized by 2 subunits. One is the S1 subunit, and another one is S2 subunit. This is the cleavage site, which means that that is where the furin cleaves this protein into 2. And you can see that each one has additional functional domains. The SP, which is signal peptide, RBD, which is the receptor binding domain, FP is fusion peptide, HR heptad repeat, and there's 2 of them, TM transmembrane domain, and CP cytoplasm domain. So what does this protein actually do? Here you can see this is the structure that we were just looking at. The structure actually has an additional region down here that goes into the membrane of the virus. And outside, you see the S1 and the S2 subunits. And the S1 attaches to host cell and shields the S2. So the S2 down here hidden underneath this cap is actually the functional component of the virus. Here you can see the whole virion. Virion is the whole particle. The particle has this important spike coming out of it, protruded, and that is why we call it the coronavirus. So the spike glycoprotein, when it comes close to the ACE2 receptor, which is in the membrane, it actually opens up. So you can see that this is the down conformation and this is the up conformation where this opens up. And on top of that, there is the fusion, receptor binding domain. And the receptor binding domain is what actually binds to the ACE2 receptor. So this is what this protein looks like. On top, you can see the S1 subunit like a cap. And on the bottom, the S2 subunit. So when we were looking at this comparison between bats, pangolins, humans, etc., we looked at a very specific region. Right? Remember that there were a few amino acids added into the novel coronavirus COVID-19. Okay, so take a look at this closely, right? This is the sequence alignment. This is that spike glycoprotein. And here in a specific position, you can see additional SPRRA sequence that does not exist in the SARS-1, bat SARS-1, or the pangolin sequences, right? So how could we know what would this have an impact? What kind of an impact would this insertion of a specific sequence have on the ability of this virus to enter and to replicate? The sequence is actually found right here on the connection between the S1 and the S2 subunit. And this is called the furin cleavage site. Now, furin is an ubiquitous enzyme, so that means that it, is in many different places that regulates many different processes in health and disease. And one key player is the ubiquitously expressed serine protease furin, which cleaves a plethora of proteins at polybasic recognition motifs. So this specific sequence of amino acids is called a motif, and this motif is recognized by this furin. And so this, different furin activity is regulated on the host side, but the viruses actually use furin, and that's why this cleavage makes this S2 subunit more exposed. So here you can see the spike glycoprotein rotating, and there are 3 chains to the spike glycoprotein, A, B, and C, and these 3 chains form this top unit. So if you cut off the top area, this is just the top. Once it's cut off, you can see that it's very simple to cut it off because it's essentially just one little cut on the chain. So what people have found by comparing these different sequences is that that area on top is much more variable. As you can see right here, this top area is much more variable than the internal area, because when we think of adaptation between the host, let's say bats or pangolins and humans, the main area that has to change is the receptor binding domain. The receptor binding domain is the specific portion of the protein that binds to the ACE2 receptor. And so this is the receptor binding domain, are these 3 little areas right here. Now, when it binds, the protein opens up. You can see here an animation of how it opens up. So this protein, when it opens up, that's when it becomes activated and can actually get connected to that ACE2 receptor. Whenever it does that, it is bound to the receptor, means that, that's the active site. But when it is closed, it is actually hiding that portion of the protein from the immune recognition, right? So there's no way to recognize that portion without it being open. So this is a complicated machinery, very fascinating how it works, because it is able to evade immune detection and hide the active part of the protein just by having this complex structure that responds to the changes in the environment that it's in. Okay, and here's the view from this side. So essentially, once the virus is close to the host cell, this is the ACE2 receptor binding domain. It comes in, opens up, and now it is in the up conformation and it is bound. And then the top part is cut off. So remember that these are the cleavage sites right here. And down here, you have the active sites. The active sites are changed. They change the way that they unfold, essentially, right? So here, they're kind of folded and hidden under this cap. Then they unfold and form this needle-like particle that allows the virus to get into the host cell. So when we think of that little insert between the human and the non-human host, that is an additional change that happened that allows the virus to be more effective at entering the cell. Now, if you remember, this is just a brief overview of the structure, right? Another aspect of how we can study these proteins is by looking at the cell response, at the host response. And in future lectures, we'll take a look at how, after the viral RNA is produced, we can study expression of what goes on inside the cell. Transcriptomics, right, will give us a sense of what types of genes on the host side are being expressed so that we can analyze what happens after the virus actually gets in. So for example, this study that you see right here is a study on the infection of SARS-CoV-2 compared to a number of other viruses that also infect lungs. And we can study in comparison how this virus is different in causing disease than other viruses, other similar viruses that are also respiratory, that cause respiratory distress. And so we'll see some of those aspects that we've heard about in the news, for example, cytokine storm, and we'll see how some of those subgenomic viral RNAs cause interference with the host factors that activate pathways to generate expression of different cytokines. And so by looking at these examples, we'll be able to understand more about the structure and the function of the different viral proteins and the non-protein coding genes. I do recommend next time that you take the time to start looking into the transcriptomics courses, because in our next series of sessions, we'll start talking about the host response to these viruses, thinking about how can we understand the biology of infection by looking at the immune response. And so different sections here in transcriptomics 1, 2, and 3 are going to be very important for us to understand how to process the gene expression data, how to do differential gene expression analysis, and then how to apply machine learning to this type of data so that we can detect subtle patterns in the response of the host. Next time when we meet, we're going to do a hands-on session. So I know that a lot of you have not yet either started the pipeline or have not completed the pipeline. So hopefully after today's session, you'll be able to go back and run some pipelines. Don't just do one, do a couple. For example, you can do a comparison of different hosts, or you can do a comparison of different strains affecting human hosts. You can even do a comparison between geographies, right? So whatever you do, if you can prepare that pipeline by next time, because we would like to look at the origins, continue looking at specific differences, and start looking at what those results actually mean when we look at results of the comparisons. Do you have any questions for me on what we covered today?
Yes, sir. This is Deirdre, Dr. Brodsky. Could you go back to the where I believe the S2 was unfolding in the yes. Okay. So we said that the furin is what is actually cleaving, or rather, when this blue component opens, the furin cleaves off the lower portion and allows the viral genome to penetrate the cell. So if we go to the next slide, where it's unfolding.
This one.
The next one. Yeah, right. Yeah, it's opening there. So, okay, it has actually, it's fooling our immune system when it's closed. So once it opens, then the next component, because this is attached to S1, correct? That
ACE2. And this is S1, yeah.
Oh, okay, wait a minute. So ACE2 is just a piece of it, is a component of the upper portion. Is that correct?
No. ACE2 is a human receptor. So this is in the human cell on the membrane. So
okay.
Let me go back to here. So here,
I kind of
can see it here. Sorry for the many different ways to represent this. This is the spike protein from the virus.
Right.
And when it comes close to the ACE2 receptor, this is the ACE2 receptor in the human cell, that's when this top part, which is the receptor binding domain, RBD, opens up.
Okay, the RBD is in what area? Is that from S2 of the virus, or
S1.
S1,
Yeah.
correct. Okay, so
So the
as
top one is S1, the bottom one is S2.
right, okay. And so now go back to the other area where we were unfolding.
So
I
this
get
one?
slide. Next one. Okay,
This?
so in this light blue is the human receptor and it's binding. Then let's go to the next one. And the next one.
This one.
Right. So I'm looking at that FP right there, the fusion of the viral with the host membranes. So now what portion of the glycoprotein is that?
That's the S2, but it got untwisted. So it's
Okay.
like a spring that is being held down by this cap on top. The cap connects to the human receptor and falls off. Well, it falls off because of the furin cutting it off. But then the inside part, right? So if you see right here, not right here, one sec. So if you see right here, right, this is the top part, comes off, and then all of this is like a spring. So it untwists and becomes this long thing that then connects the two membranes together.
Okay, very good. Okay, I've got it now. I just saw a couple of parts and I wasn't getting it all together. So that is in fact S2 that is unfolded.
Yep.
Yes, thank you.
All right, any other questions? Yeah, go ahead.
Can you hear me?
Yeah.
Okay. Can the environment you're in affect like your susceptibility to coronavirus?
The environment?
Yes, because like it originated in China. It originated in China, which has like 2 billion people, compared to the U.S., which has 300 million people. So like, it's more of a crowded environment, as well as like little everyday things like diet and genetic predisposition. So like, can all those things, like those three things, like with the crowded environment, your genetic predisposition, and diet, like can they play a bigger effect? Can they play a big effect on like your susceptibility to COVID-19? And like, if they can we like, is there a way that we could like unravel that with with our database?
Yeah, so I think that that's a great question. I think many people are asking the same question, and many people are saying, you know, could it be environmental factors like heat and humidity, which affect the viral structure and things like that? Could it be, like you said, genetic factors, the differences between Asian people or the diets that they have as compared to us? So all of those factors theoretically should have some effect, but no one really has a conclusive study on some of these factors. There have been some studies, for example, it is known that men are more susceptible, it seems like, than women, or younger people have, you know, less infections than older people. Or, for example, there have been studies about smokers. This ACE2 receptor seems to be more present in people who do smoke than in others that don't smoke. But those types of questions, like you said, could be asked of data. So what's very important about this is that there's a whole lot of publicly available data sets. So if you look on NCBI, and that's exactly where I was trying to get to with this, you know, next series of sessions that we have, you can look at, you know, COVID, for example, or you can even look at ACE2, right, which is the receptor. And you can look at, for example, gene expression omnibus is a database for gene expression, where you can look at a variety of different data sets that allow you to ask those kinds of questions. For example, you can say, okay, is there a mutation in the ACE2 receptor that is unique to, let's say, the Asian population or the African American population or the Estonian population or any other kind of ethnicity? Or is there a difference in level of expression between different groups of people? For example, maybe some have more of that gene expressed and others have less of that gene expressed. Some of those questions could be answering some of your questions. So here's a data set, for example. SARS-CoV-2 receptor ACE2 is an interferon-stimulated gene in human airway epithelial cells and is detected in specific cell subsets across tissues. So maybe it is expressed differently in different tissues. So getting from that kind of here you have in vivo antiviral host response to SARS-CoV-2 by viral load, sex, and age, right? So that's exactly some of those aspects that we talked about. I think it's worthwhile to look into these data sets to see what you can find. I think that there's a variety of factors that probably play a role. I do want to say that I'm not an expert on coronaviruses, but there is somebody that I know that is. And we recently gave a presentation together. It's a little bit long, but it is worthwhile to listen to the first part of that presentation that talks about the molecular biology of SARS-CoV-2 by Dr. Gus Kasoulis. I'm going to post that on the forum as well for all of you to see. Okay, any other questions that I can answer?
Sorry.
Yeah.
This is another question that occurred to me. Like, before, like when the coronavirus was like very, rampant, like old people were basically more susceptible to it because of a weakened immune system. When we use NCBI with bioinformatics, like, can we pinpoint exactly like when exactly their health fell off, or can we pinpoint like what gene is responsible for that weakened immune system, for something like a weakened immune system, or diseases like heart, or any disease really?
Well, yeah, look at this data set, for example. We examined host gene expression across infection status, which is, how severely infected they are, viral load, how much virus do they have, age, and sex, among RNA sequencing profiles of nasopharyngeal swabs from 430 individuals with SARS-CoV-2 and 54 negative controls. SARS-CoV-2 induced a strong interferon-driven antiviral response and reduced transcription of ribosomal proteins, etc. Right? So you can actually now look at this data. They have 484 data sets or data, samples. So you can definitely do that kind of an analysis, or you can read the paper that they published here if you would like to, just kind of, well, they haven't published the paper yet, but you can definitely look into those questions. I don't have a definitive answer about that, but you're welcome to look at these data sets.
Well, and I'm sorry to interrupt. Also, I have heard that there was, let's say, in particular hotspots when there were higher concentrations of virus, let's say, in St. James Parish, and they also attributed that increase to not only the health of the African Americans that were present there, it was also including information as such that regarded the pollution or the pollutants that were in the environment. So
right,
that
so
was interesting.
Another great point is that there's probably environmental factors that play a role as well. So we don't have all of the answers just by looking at the genomic sequence of the virus, and we don't have all the answers just by looking at the it's really a complex complex interaction of many different factors. I think it would be useful to look at this recording and post it on the forum. You know, then when we go back, right, we can start thinking about what types of data do we have access to? That's question number one. Number two, what kind of analysis can we perform to get answers to these questions that you have? And probably to make the questions much more specific as we think about the issues at hand, right? So when we say, okay, could it be sex? Could it be environment? Could it be pollution? Could it be, ethnicity? All of these different factors, right? Yes, there's a lot to figure out, but could I focus my question on one particular element of that, story, and say, okay, how exactly does the immune system respond to viral infection? Or how exactly does it respond differently between males and females, or African Americans and Hispanic, or white people? So we can start making those questions much more specific, and that is the objective of this program: to learn enough about methods of analysis, different data sources that are available, and start formulating that question that you could potentially spend a couple of weeks doing the analysis and coming to those conclusions on your own, to be able to make that your own conclusion and your own analysis, that that you're basing your understanding on the data that you found and you've analyzed. All right, well, I have to go to my next meeting, but thank you all for joining today. I posted the webinar right here. Please take a look. And if you have any other questions, or you're starting to think of project ideas, which is really good, I'll start putting up some direction on how to think about your own project and how to formulate that question and find the right data set in our next session. And please start working on transcriptomics. That will help you in the few next sessions that we'll do. All right, take care, everyone.